Cell-to-cell communication is vital for living beings to respond and function. It is how we provide nutrients to cells and send signals across the body to start certain motor functions we may want. To understand the basics of communication, we will delve right into the simplest bits. There are multiple types of communication, but there are two basic ones, and they are paracrine signaling and endocrine signaling. Paracrine signaling describes short-distance signaling, where a signaling cell immediately sends out signal molecules into the nearby extracellular space. These signal molecules, also known as paracrine factors, are then picked up by nearby cells. An example of paracrine signaling is neurotransmission. Endocrine signaling describes long-distance signaling, where a signaling cell immediately sends out signal molecules, also called endocrine factors, that are sent out into the bloodstream to a target cell. This is how hormones are sent out to the body. Animal cells have their signals sent out into their extracellular matrix, and this matrix is full of protein and largely collagen mixed with proteoglycans, which help with communication between cells. These cells can communicate through tight junctions, gap junctions, and desmosomes. Tight junctions are water seals between two adjacent cells. Gap junctions are channels between two cells for the transport of nutrients, and desmosomes are spot welds between two adjacent epithelial cells. Plant cells communicate through plasmodesmata, which are channels between two cells that connect to their cytoplasms because of their cell walls which prevent normal communication. The signal molecules are also known as ligands. Ligands begin the cell transduction process after binding to the receptor protein. Signal transduction is the process of how a signal molecule is processed and sent throughout the cell, which can be divided into three stages: reception, transduction, and response. Response is when the signal molecule binds to a receptor protein on or inside the cell, which changes the receptor's shape. Transduction begins inside the cell, where a series of relay molecules send a message down to the nucleus, resulting in a cascade effect. Response is when the cell changes its behavior based on the message. There are four general types of receptor proteins: G-protein linked receptors, tyrosine kinase receptors, gated ion channel receptors, and intracellular receptors. Since epilepsy deals with neurons, we will talk about gated ion channel receptors, the simplest to understand. Neurons use glutamate receptor channels, GLRs, which is a type of ligand-gated channel receptor, which are receptors that open from a ligand. When a neuron has a glutamate ligand bound to a receptor, it opens the gateway of the neurons, and once it is gone, it closes. How epilepsy messes with this process will be discussed later in this video. During signal transduction, there is a process called a kinase cascade using protein kinase. A protein kinase is an enzyme that modifies other proteins through the means of phosphorylation by a covalent bond. A protein kinase cascade is when the activation of one protein begins the process of the activation of the same proteins in the next tier. The signal is transmitted onto intracellular downstream proteins by protein phosphorylation and activation of cascade components. Specific effector molecules and downstream cellular responses are activated. This helps the original signal to become amplified and spread throughout the cell. Cyclic AMP, or CAMP, and calcium ions are these second messengers that begin the kinase cascade. During signaling, ligand-gated calcium ion channels like calcium flow into the inside of the cell, and depending on the concentration of cytoplasmic calcium ions, can include different types of cell responses, muscle cells contracting. For CAMP, it is synthesized by enzyme adenylyl cyclase from ATP. Cyclic AMP binds and activates to CAMP-dependent kinase, A kinase, which regulates vital metabolic pathways, phosphorylates serine and threonine residues of its target proteins and activates them in the process. Cyclic AMP are found in many cells and have different target proteins and therefore responses. Upon receiving the signal after signal transduction, it typically can lead to gene expression, using information from a gene in order to produce a functional product, which is typically a protein. Cellular metabolism is also a gateway where metabolic enzymes are either more or less activated, and apoptosis is also communicated through the signal transduction system. Epilepsy is not a single condition. rather a collection of conditions that function the same under one unifying term. There is also no single way of inheriting epilepsy, as anyone at any age can get through epilogenesis. Epileptogenesis describes the gradual process through which a previous non-epileptic brain undergoes pathological changes that ultimately lead to the development of epilepsy. Structurally, epilepsy can occur due to abnormalities in the anatomy of the brain that can increase the risk of seizures, which are strokes, traumatic brain injury, brain tumor, or central nervous system infection, including genetic/developmental/developmental origin. Ociotemporal sclerosis is a common cause of temporal lobe epilepsy. Infections that target the central nervous system can directly affect brain tissue and lead to long-term seizure susceptibility, like herpes simplex encephalitis, cerebral malaria, toxoplasmosis, toxocariasis, if I said that right, and neuropsychic psychosis. Not gonna try with that one. The immune system can also attack the brain tissue, which is autoimmune encephalitis. Celiac disease is also associated with epilepsy in rare syndromatic forms, like the triad of epilepsy or the Lennox-Gastaut syndrome. Metabolic disorders can disrupt the brain's normal function and could cause epilepsy to occur. In rare cases, it can be inborn errors of metabolism, like mitochondrial disease, urea cycle disorders, or glucose transport 1, type 1, GLUT1 deficiency. Even with all that, there are still many cases of epilepsy that have an unknown origin as well. Epilepsy, genetically caused, is mainly an autosomal and dominant trait. However, in rare cases, it can be a recessive trait or sex-linked as well. The following list is of affected chromosomes and the type or trigger of epilepsy that goes with it, both voltage-gated and ligand-gated. Chromosome 2, 2q24, harbors SCN1A, 2A, and 3A genes, and those mutations are related to Dravet syndrome and generalized epilepsy with cerebral seizures plus, or GEFS plus. Chromosome 20, 20q13, harbors KCNQ2, major cause of benign familial neonatal convulsions and other infantile epilepsy syndromes. Chromosome X, where many X-linked epilepsies occur, such as ARCS, West syndrome, CDKL5, Rett syndrome variant, and PCDH19, epilepsy in females. Chromosome 15 in 15q11, q12, and q13, associated with Angelman syndrome and Dup15q. Chromosome 8, 8q24, harbors KCNQ3, which works with KCNQ2 to regulate neural M currents. Chromosome 6, 6p12 to p21, of juvenile myoclonic epilepsy, with EFHC1 and BRD2. Chromosome 1, with CHRNB2, nocturnal frontal lobe epilepsy, and SLC2A1, GLUT1 deficiency. In chromosome 3, CLCN2, which is with juvenile absence and myoclonic epilepsy. In chromosome 10, LGI, autosomal dominant lateral temporal lobe epilepsy. Chromosome 12, TUBA1A, and WDR62, which is lissencephaly and polymicrogyria. Chromosome 16, PRR2T, self-limited infantile epilepsy, and TSC2. Chromosome 17, LIS1, which is also lissencephaly. Chromosome 22, DEPDC5, familial focal epilepsies. Which that whole list might not even be complete. All these mutations have one thing in common. That is, they all relate to the production of an excessive neurotransmitter production, which is primarily glutamate. The excess glutamate results in excitotoxicity, where nerve cells suffer damage or death at levels of safe neurotransmitters become pathologically high, resulting in excessive stimulation of receptors. This happens due to the high levels of calcium that enter the cell, which activates a number of enzymes that damage cell structures. Epilepsy is characterized by a long-term tendency to experience recurrent, unprovoked seizures, the main syndrome of epilepsy. Seizures are described as excessive and rapid non-firing in the brain. Seizures are grouped into four main classes: focal, generalized, unknown whether focal or generalized, and unclassified. Focal seizures originate in one area of the brain and may involve localized or distributed networks. They are split into states of consciousness to whether the affected person is aware and/or responsive. An experience, or aura, precedes focal seizures, and these seizures can include sensory, psychic, autonomic, and motor phenomena. Muscle jerks may start in specific groups and spread to surrounding muscle groups. Automatisms may also occur, which are non-consciously generated activities. Focal seizures can evolve into focal to bilateral tonic-clonic seizures. Generalized seizures originate at a specific point and quickly spread across both brain hemispheres through interconnected brain networks. Forms can include generalized tonic-clonic seizures, absence seizures, or other generalized seizures, tonic-clonic, myoclonic, atonic seizure, and epileptic spasms. Trigger seizures can be activated by sleep deprivation, stress, fever, illness, menstruation, alcohol, and certain medication. Reflex seizures can be provoked by specific stimuli, such as flashing lights, sudden sounds, or specific cognitive tasks, such as reading or performing calculations. Some people, depending on their type of epilepsy, may experience seizure clusters that give them the worst quality of life. It is described as either 2 or more seizures per 24 hours or an increase in seizure frequency over a person's usual baseline. Postictal states are periods after seizure where there is a period of recovery during which there is confusion. It can be noticeable by confusion, headache, fatigue, or speech and motor disturbances. Epilepsy can affect the psychological and social well-being of an individual, where that individual may experience social isolation, stigma, or functional disability. Neurodevelopmental and psychiatric disorders are also more common in individuals with epilepsy. That is, depression, anxiety, obsessive-compulsive disorder, and migraines. As well, ADHD is prevalent among children with epilepsy, and in general, with children in the autism spectrum disorder.